Welcome to section 21.6b. Okay, gentle people, we're going to go ahead and continue our discussion on biopolymers. The next biopolymer that I want to talk to you is the DNA molecule. So, like our proteins, we're going to first talk about our monomers. Now, the monomers to build the DNA polymer are going to be called nucleotides. And here is the general structure of a DNA nucleotide. What you will have is a phosphate group, and this is just a phosphorus surrounded by oxygen atoms. Now, connected to that phosphate group is a sugar, and we'll talk about what constitutes and what classifies sugars. For now, what I want you guys to know is that this sugar is a 5-membered ring with 4 carbons right here, and it has alcohol groups that are attached on that sugar itself. The last thing a DNA nucleotide is going to contain is a nitrogen base. And so this base is going to be made out of nitrogen, carbon, and hydrogen. This is called the nucleobase. Now, you might not have thought of DNA as a polymer, but in fact it is. So here's what happens. I'm going to take a DNA nucleotide and I'm going to combine it with another DNA nucleotide. And the way that I'm going to do this is another condensation reaction. Now, this condensation reaction isn't something that we've talked about before, but it follows the same principles. What I'm going to do is I'm going to take an H from my alcohol, and I'm going to combine that H with an OH off my phosphate group of another DNA nucleotide. Now, usually what's going to happen is a catalyst is going to be used. Ligase is usually the one that's going to be used. And what's going to happen is you're going to go ahead and form a connection or a linkage between the phosphate group and a sugar off 2 different DNA nucleotides. Now, what you guys will notice is that after I do this reaction, I still have another alcohol group. So that means I can continue adding DNA nucleotides this way. And on the other end, you see that you have a phosphate group that can continue to add DNA molecules on the other end. So what this does is it goes ahead and puts all these phosphates in a row, and I polymerize all these phosphates. This is what's called the phosphate backbone. Now, off of this phosphate backbone, what you're going to have are the nucleobases pointed in one direction. And so this makes one strand of DNA. Now, if you ever studied DNA, you know that this is made out of 2 strands. So let's take a look at those nucleobases. Now, you guys might have learned this in high school. There are 4 nucleobases here on Earth, and they are thymine, adenine, cytosine, and guanine. And they are usually abbreviated T, A, C, and G. So what happens is that when you get one strand of DNA, you get these bases pointed on the inside. So let's say T, A, C, and G. And what's going to happen is that the second strand of DNA is going to come and it is going to match these bases up. T is going to go with A and G is going to go with C. And this builds us our double strand. Now, if you've ever wondered why T is only paired up with A and why only C pairs up with G, well, now we can explain it to you. The reason that thymine matches with adenine is because of hydrogen bonds. What you guys will see here is on thymine, I have the configuration acceptor and donor. And what you'll note is that adenine is the exact opposite. It has donor and acceptor for my hydrogen bonds. And so that's why these 2 will come together. If I look at cytosine, we have donor, acceptor, and acceptor, where guanine is going to match for hydrogen bonding. We have acceptor, donor, and donor. And that's why these bases come together as pairs, because they match each other through hydrogen bonds, and you will very rarely get mismatches in these base pairings. Now, one thing that is famous about DNA is not only is it double-stranded, but it has this helical twist to it. So let's explore why there's a helix in DNA. So to do this, I'm going to use a different little website that builds molecules for us. So let's start out with one block. So remember, DNA is a polymer made out of monomers, where the monomers are the nucleotides. So here's my block, and I can go ahead and rotate it around. So what I told you guys about polymers is we are stacking one monomer on top of each other. So what I can do is I can stack multiple blocks on top of each other, and if I were to do this, what you would get is this nice stack. And so this doesn't look like a helical structure. Well, in truth, our polymers aren't these perfectly rectangular objects. Instead, you can envision them as wedge-shaped blocks. So in this case, you can think of a box where one end is tapered. So let's go ahead and see what happens if we go ahead and stack wedges on top of each other. What you guys will see is that I kind of make a donut structure where it comes back on each other. So this isn't the structure of DNA that we're normally used to. What we should note is that when I make my polymer chain of DNA, it is not only wedge-shaped blocks, but there's a slight displacement on when I stack those wedges together. So in fact, the wedges are not one on top of each other. They're slightly displaced. So let's go ahead and stack these together. So if I were to stack them together, what you guys will see is that I start to build my helical structure. So this would be one strand of DNA. Now, if we wanted to go ahead and get a better representation of our DNA molecule, we can go ahead and build one strand of DNA. So these are the nucleotides built one on top of each other, giving me a single strand. And so let's go ahead and match the second strand on top of this. And so this is the structure that you guys might have been used to. This is the helical structure of DNA with two strands where my bases are binding those two strands together. If you guys want a short little break and aside and you're really interested in the DNA molecule, you guys can look up this journal article. It was a really neat article that was written about 6 years ago, and what they did is they made the first synthetic DNA nucleotides. So as I mentioned before, on planet Earth, we only know of 4 DNA nucleotides, A, T, C, and G. What these chemists did is they made 2 more nucleotides, X and Y. And if you guys take a look at X and Y, they are made out of non-polar things. They don't hydrogen bond at all. And so these two will pair up. The researchers went ahead and they incorporated this into a living organism. They were able to have that organism replicate X and Y into part of its DNA. Okay, Geno people, we're going to go ahead and shift gears, and we're going to talk about carbohydrates. Carbohydrates are a molecule that has a whole bunch of energy packed into it. This is what biology uses to store potential energy and later use that energy. Now, if we think about its name, carbo means carbon, and hydrates means something with water in it, so H2O. So the empirical formula for carbohydrates are CH2O. Now, the number of carbons can vary. 3, 4, 5, and 6 are the most popular in biology. And when we make a carbohydrate, we usually call them monosaccharides. Based on the number of carbons it has, we classify what type of monosaccharide we have. So if it has 3 carbons, we call that a triose, 4 tetrose, 5 pentose, and so on and so forth. Another name for monosaccharides are simple sugars. If I take two sugar molecules and combine them, instead of a monosaccharide, I will have a disaccharide. And as you guessed it, I can combine more than two sugars together or more than two monosaccharides, and I will get a polysaccharide. So what you guys can glean from this is my monosaccharides are going to serve as my monomer, and I am going to polymerize them to make biopolymers. Now, one important classification on monosaccharides is what groups they possess. The two major classifications for monosaccharides or simple sugars are aldoses and ketoses. And this is based on the functional group that they have at the end of the molecule. Now, an aldose is going to have an aldehyde group at the end. And to remind you guys, an aldehyde is this functional group. They can also be classified as a ketose, and a ketose is going to have a ketone functional group. So again, to remind you, this is what a ketone is going to look like. Now, oftentimes you are going to see the carbohydrates presented to you in this fashion. And that means that I'm just going to string my carbon chain all down the line, and I'm going to put the OHs, the aldehydes, on either side of this string of carbons. Now, in organic chemistry, they'll teach you what it means to be on the left and right side. But right now, what I want you to do is be able to tell me if carbons are chiral or not. And this is going to be important later down in organic chemistry. So to do this, let's go ahead and start with an eye-clicker question. Look at fructose right here, which is considered a monosaccharide, and tell me how many chiral carbons this has. So I'm going to give you guys a little trick when doing chiral carbons in monosaccharides. First, let's talk about sp3 carbons. Now, for an sp3 carbon, what you're going to have is carbon, and it's going to be tetrahedral. So that means line, dash, wet. Now, let's say that this carbon has 4 different things around it. Now, we've already seen this type of molecule. If I give you a tetrahedral with 4 different things around it, then you know that this is going to be chiral. You guys can go back to that lecture and see the example that I gave you, which was the same exact example. Now, what we can do is we can go ahead and play the what if game. So in the first case, I have 4 different things. Let's say that I have 3 different things on my second scenario. So you guys can test this out yourself. This actually happens to be achiral. So let's do another case. And as you guys might have guessed, this time we're going to put only 2 things that are different around it. And again, this is going to be achiral. And obviously, if you put the same thing around it, it's going to be achiral as well. So the take-home message here is that a tetrahedral can only be chiral if there are 4 different things around it. Now, the other thing that we have to talk about with sugars are sp2 carbons. So an sp2 carbon is going to be trigonal planar. So let's go ahead and put 3 different things around it, and you guys can test it yourself. This is achiral. And we can go ahead and put 2 of the same things, 3 of the same thing. They will always be achiral. So let's go back to our iClicker question and use this knowledge. So here's our first carbon. If I look at that first carbon, what I will notice is it is sp3, but there are 2 hydrogens on it. So if that's the case, that means that this carbon here is achiral. The next carbon that I have in fructose is sp2. Now, we already said that sp2 is not going to be chiral, so again, this carbon is achiral. Now let's get to the third carbon here. Now this third carbon right here, let's go ahead and see what's around it. I have an H on one side, I have an OH on one side, and on top, I have a carbon connected to it. Now that carbon chain is 2 carbons long. It has this carbon and this carbon going up at the top, so I'm going to call that R1. Now on the bottom, there's a chain of 1, 2, 3 carbons. So even though there are carbons on the top and bottom, they are different carbon chain links, so that means I'm going to have to specify that it's something different. So I'm going to call this R2. So if I take a look at this carbon is sp3 and it has 4 different things around it. So that means that that carbon is chiral. So to designate a chiral carbon, I'm going to put a star around it. So let's take a look at the next carbon down our chain. So for this carbon, we see that it is sp3 again, OH, H, and if I look on top, I have a chain of 1, 2, 3 carbons. So I'm going to call that R1. And on the bottom, 1 and 2. So this is a different R group on there. So again, 4 different things around an sp3 carbon. So that carbon is chiral as well. So I'm going to let you guys do the next couple 2 by yourself. I'll tell you guys the answer. You guys will note that this next carbon right here, it has 4 different things around it. It is sp3. And so that is going to be a chiral carbon. This carbon right here, even though it has sp3, it has 2 hydrogens on it. And so something that is sp3 with 2 of the same things on it is a chiral. So in this molecule, I have 3 chiral carbons, and they are starred for you guys. So let's go a little bit more in depth on carbohydrates. So this is the form that carbohydrates are usually given to you guys, but I'm going to tell you that this is not the most stable form of carbohydrates or simple monosaccharides. What happens with monosaccharides like fructose is they tend to cyclize in biological media. So what happens here, instead of my carbohydrate being this long line, it's going to wrap around itself and it's going to react with itself. What's going to happen is the alcohol group is going to interact with the carbonyl carbon. And what's going to happen is the hydrogen is going to pop off my alcohol, go to the carbonyl carbon, make a new alcohol, and while it does that, the carbonyl carbon is going to form a bond to that oxygen. And the end result is I get a cyclized monosaccharide. Now you might be asking yourself, why does this matter? Well, this is the form that the monosaccharides are going to start to polymerize. So here are my cyclized structures, and what you guys will see is I have alcohol groups. Now, these alcohol groups are going to condense with each other. So that means I'm going to take an OH and an H, and I'm going to put it together, and I'm going to take out water. And then what's going to happen is I'm going to form a bond with the rest of the monosaccharides. I'm going to get an oxygen to go ahead and link up to one of the carbons. This is called the glycosylide linkage. So it's just like every other condensation you've seen before, where I'm going to go ahead and bring 2 monomers together. Now, when I take 2 monosaccharides and I polymerize them, I get disaccharides. And these are ubiquitous in nature. You see them all over the place. If I take glucose and fructose and I take these 2 different monosaccharides and combine them, what I get is something called sucrose, and you guys know this as common table sugar. If I were to take galactose and glucose and put them together, what I get is lactose. And so for those of you guys that are lactose intolerant, this is the devil for you guys. Now, the reason I'm bringing up lactose is what you will note is that these disaccharides have a lot of potential energy. The carbohydrates is how biological organisms store energy. And lactose has a lot of energy stored in it. However, for those of you that are lactose intolerant, you lack an enzyme to break lactose apart. You see, what happens in your body is you're going to take the disaccharide and you're going to use enzymes and add water to break them up into the simple sugars, where then you can further metabolize them. For those of you guys that are lactose intolerant, you lack the enzyme to break this linkage. So what happens is that lactose passes through you, gets into your stomach flora or gut flora, and the bacteria there, they see the lactose and they are able to metabolize it, and then you end up with a bad time. And this is what biology does with simple sugars. These simple sugars store a lot of energy, so I do not want to get rid of these simple sugars. So what biological organisms do is they polymerize these simple sugars and store them. So I can go ahead and take a simple sugar, combine it with a simple sugar, making that glycosylide linkage, and I can do this over and over and again. Plants can store sugars in this straight chain, and if they go ahead and use glucose and put that glucose in a straight chain, we call that amylose. It turns out mammals like us also take glucose, but we like to go ahead and make that glucose branched. And so instead of a straight chain polymer, my polymer branches off here and there, and this is called glycogen. Now you might be asking yourself, what is the benefit of polymerizing these monosaccharides? Why don't we just store the monosaccharides as individual monomers? Well, it turns out if you polymerize these monosaccharides, you reduce the osmotic pressure in your cells. So let's take a look at why this works. Let's say you have 100 glucose molecules. Let's say you go ahead and polymerize those glucose molecules and you make one polymer, and let's say that this is glycogen. So this is off of a mammal. Okay, so let's go ahead and calculate osmotic pressure. Our osmotic pressure equals IMRT. And so both of these things are not going to break up once I put them in water, so I is 1. Let's go ahead and say that R and T, you're doing the same temperature in your body, and the gas constant isn't changing. What you can say is the osmotic pressure is dependent on molarity. And so remember what molarity is. That's going to be moles over liters. And so as the number of moles go up, so does the osmotic pressure. And so take a look at my chemical equation. I start out with 100 things, and then I go to 1 thing. And so what that means is the number of moles is being reduced. I'm going from 100 moles of glucose to only 1 mole of glycogen. And so that means I'm going to drop the osmotic pressure by 100-fold. And so this is the way that we can store these highly valuable energetic particles and not burst our cells open. All right, Chem 1C, I hope that made sense, and remember to stay safe.